welcome. My name is Yolande John. I'm the meetings manager here at ASPIT. Thank you for joining us for this Focus on Pharmacology session, where today we welcome the Division of Drug Metabolism and Disposition for their 8th installment in their quarterly series, Current Advances in Drug Metabolism. Before we start, just a few housekeeping items. You will note that all attendees are on listen mode only. Some of you have already found the chat function where you can send messages to each other. If you have any questions for the speakers, please use the Q&A function that can be found at the bottom of the window or on the desktop or the top of a tablet. You can submit questions throughout the presentation. You can even upvote questions you like by using the thumbs up icon. By attending this session today, you consent to ASPIT's Code of Conduct and having your questions and chat recorded. A recording of today's session will be made available in ASPIT Connect. We will send out notifications once it's ready. You'll also receive a survey link from us that should take about 2 minutes to complete. As always, we look forward to your feedback. Okay, let's begin our session for today. I would like to introduce our moderator, Dr. Young Jeong from Purdue University. Dr. Jeong, welcome.
Thank you, Yolande. Welcome, everyone. This webinar series is to stimulate exchanging ideas and promote communication among DMDD members, and also to highlight the career development of young scientists in the field of drug metabolism and disposition. With that, I would like to introduce our first speaker. As usual, we have 2 excellent speakers, and our first speaker is Dr. Jed Lam. Dr. Lam received a PhD in medicinal chemistry department from the University of Washington, and then completed postdoctoral training at UC San Francisco in the lab of Dr. Ortiz de Montellano, where he studies cytochrome P450 ligand binding dynamics and their relationship to enzyme function by using 2-dimensional NMR techniques. He is currently an associate professor in the Department of Pharmaceutical Sciences at the University of Colorado, Skaggs School of Pharmacy. His current research interest is in understanding and predicting drug metabolism and disposition in special populations, including neonates, pregnant people, and also cystic fibrosis patients. He's currently serving on a number of editorial boards, including Nature Scientific Reports, Current Drug Metabolism, and PLOS ONE. And with that, the floor is yours, Dr. Lam.
Awesome, thank you. Can everybody hear me okay? Okay, I see thumbs up. All right, great. Let me start sharing screen. Oh, that's not start. There we go. Does everybody see the Rocky Mountains? Hopefully. Great, wonderful. Well, I just want to thank you, Young, for the invitation and the great introduction. Also, Carrie and Clarissa for helping organize this, and of course, Yolande and Aspect for having these. I found these seminars really wonderful opportunities to get to know each other and to share some great research. I hope we continue them in the future. Okay, so today I'm going to talk to you about the role of CYP3A7 in parturition in adverse drug hormone interactions. But before I do, if you can see this is the view I get to see on the way to work in the morning. So Colorado is also known as the Rocky Mountain State, right? So we have more 14ers, or that's mountains above 14,000 feet tall, than any other state in the country, including Alaska. There's 53 of them here. So if you ever get a chance to visit Colorado, you should take in some of the scenery. All right, so brief outline today. I'm going to talk to you about the metabolic changes that take place in utero and during the first year of life, and really how they impact drug metabolism, disposition, and toxicity in these special population groups, including fetuses and neonates. Specifically, the role of CYP3A7 in fetal neonatal drug metabolism, and also what we're defining now as its role as a fetal placental communication conduit. And I'll explain what we mean by that. I'll talk about adverse outcomes of CYP3A7 inhibition, our work with Calatra, which is a lopinavir-ritonavir anti-HIV cocktail as an example, and then talk about antiviral drugs as a source of these potential drug-hormone interactions. We'll also talk about the effect of HCV antiviral compound paratiprevir on CYP3A7. And then I'll finish with some of our really new work looking at antiviral inhibition of CYP3A7 and its impact on bile acid metabolism. And then I'll just summarize at the end. Okay, so one of the questions that keeps me up at night and that we think about a lot in my lab is, can we treat babies like little adults when it comes to drug metabolism and disposition? And you might already know the answer to this question. Of course, a drug that we're going to give this young lady here is probably not going to be metabolized, disposed of the same way as it would be for an infant. But it's important to realize that there's this continuum, right, between the fetus, the developing infant, the neonate, and then the developing infant, and you have physiological and anatomical changes occurring during that whole span. So that, for instance, a fetus is not the same thing as a one-day-old in terms of drug metabolism disposition, and the one-day-old's very different than the one-week-old, and that's very different than the one-month-old. So it becomes extremely difficult to predict how drugs are going to be metabolized in these types of patients. And in fact, Ron Hines provided a very nice table a few years back. This is from Clausen's toxicology textbook, probably a lot of you are familiar with. You can see in this table what he's done is he's broken down the enzymes that change over the lifetime of the infant from neonate out to 1 year into 3 different classes. And I might mention here, you also see changes in transporters, drug transporters that can affect disposition as well. They're not characterized here, but those can be just as important depending on the drug that you're looking at. So we have 3 different classes. Class 1 are drug metabolizing enzymes that are only present in the fetal and neonate and developing infant. Class 2, we see develop throughout the lifespan, and then class 3 are only present in the adult, so 2 years out and on. So you can imagine, this is a very complex stew of different metabolic enzymes that are changing over the course of the lifespan, going up, going down, appearing, disappearing, and that can make a profound impact on drug metabolism and disposition. Now, in my lab, we focus a lot on CYP3A7 because, of course, the 3A pathway is responsible for metabolism of about 60%, 50 to 60%, depending on the year, of known small molecule drugs on the market. So a very important metabolism pathway, both in the infant and neonate and in the adult. But 3A7, of course, is only expressed typically in the neonate and developing infant. What else do we know about the function of CYP3A7? As I mentioned, it's primarily expressed in fetus and neonates. It shares about 88% identity with CYP3A4. So it metabolizes a lot of the same substrates and produces a lot of the same metabolites as CYP3A4 does. It metabolizes both drugs and endogenous hormones. So a couple of the important endogenous hormones that it metabolizes are DHEAS and retinoic acid. I'm not going to talk about retinoic acid today, but DHEAS is important because it's the highest concentration of circulating steroid hormone in your body, and it's the precursor for all the downstream androgens and estrogens that are produced in the adrenal gland. And really important in distinguishing metabolism and metabolic characteristics from CYP3A7 versus 3A4, 3A5, for most drug substrates, they are metabolized by both enzymes. The KCAT is about 1/100th that of CYP3A4 when you're looking at metabolism by CYP3A7. So it's a much slower enzyme at turning over substrate than CYP3A4 or CYP3A5 does. Now, thanks to the diligent efforts of our collaborator, Emily Scott at the University of Michigan, we were actually able to get a crystal structure for the first time with a bound ligand, at least a substrate ligand in the active site. And here I'm showing you DHEAS. You can see it's labeled in cyan in the active site, just right above the heme iron. Interestingly, in this particular construct, there were also 3 other copies of DHEAS bound to the enzyme, and some of them in important positions that have been established previously, like the FG loop region that we think are involved in allosterism and allosteric kinetics in the enzyme. So we're exploring what's the functional significance of that. We don't really know yet. That's a story to be continued, but pretty interesting. Anyway, this gives us a good model on how a substrate can dock into the active site of CYP3A7. So what's the significance of CYP3A7 in fetal development and parturition? As I mentioned previously, CYP3A7 metabolizes DHEAS to 16-alpha hydroxy DHEAS, and then this is exported to the placenta and ultimately converted to estriol. Why is estriol important? As it turns out, estriol is the highest circulating hormone during that last trimester of pregnancy. And women with low estriol levels tend to give birth preterm to infants of lowered birth weight and that have reduced adrenal insufficiency. So what's happening here is you have DHEAS produced in the fetal zone of the adrenal glands, exported to the fetal liver. There it's hydroxylated by CYP3A7. That goes on to be desulfated, and eventually in the placenta, it's converted by CYP19A1 aromatase into estriol. So really, CYP3A7 is functioning as a fetal placental communication conduit during pregnancy. It's like a telephone, but of course it's a chemical telephone in this case, right? So I've got an old school, probably the students today don't recognize what this is here, but it is a telephone receiver, trust me. In this case, that's representing DHEAS. So DHEAS then is oxidized by CYP3A7. That serves as a signal to the placenta and the mom that everything's good to go. Estriol levels are high. We can continue this pregnancy to full term. When you have inhibition of CYP3A7, though, by a drug or other xenobiotic agent, then you get lower DHEAS oxidation, low estriol levels, and that's going to lead to premature birth and low birth weight. Why that is important is because that's the leading cause of infant mortality in the US. So it's a huge issue, and the telephone is ringing off the hook at this point. So the fetus is trying to tell the mom, hey, not all is not well here. We need to stop this pregnancy and get the baby out as quickly as possible. Drug hormone interactions can really be detrimental to the developing fetus. So interruption of this fetal placental communication conduit can lead to low birth weight, premature birth, adrenal insufficiency, as I already mentioned, and an increased risk for drug-drug and drug hormone interactions. So you can imagine CYP3A is the main clearance pathway of drugs in the neonate developing infant, just as it is in the adult. And so if you inhibit that pathway and your patient's on multiple CYP3A substrates, which they are typically in the NICU, then you can also have drug-drug interactions. And more recently, evidence has pointed to the risk for obesity and dyslipidemia later in life. And I'll talk about why we think that's going on at the very end. Okay, so a couple of years ago, we did a study where we were looking at the drug cocktail Caletra. So that's ritonavir-lopinavir combination. And so these are HIV protease inhibitors. And in fact, lopinavir is the active component of the cocktail, and ritonavir is just included as a pharmacoenhancer. So its sole function there is to inhibit CYP3A metabolism so you can keep those lopinavir levels high enough. And you do have to keep the antiviral levels pretty high in the micromolar range to be effective to have effective viral suppression. So it turned out that Caletra given to pregnant women led to infants with low birth weight and adrenal insufficiency and sky-high DHEAS levels. So this really suggested to us that you're seeing inhibition of CYP3A7 DHEAS hydroxylation, low estriol levels, which would account for those pathological effects that we see. So both lopinavir and ritonavir are inhibitors of DHEAS oxidation by CYP3A7, although ritonavir is a much more potent inhibitor. And in fact, if you look to the left here, you can see in that upper panel, what I'm showing you here is clearance study of lopinavir by CYP3A7, recombinant CYP3A7 and recombinant CYP3A4. The CYP3A7 is shown by the open squares, CYP3A4 is shown by the open circles. You can see in the absence of ritonavir, CYP3A4 is clearing the drug very rapidly. So it clears lopinavir very rapidly in the absence of the pharmacoenhancer. Whereas in the absence of ritonavir for CYP3A7, we don't see as rapid clearance, right? In fact, the clearance is very similar to what we see with CYP3A4 in the presence of ritonavir, suggesting that maybe we don't even need ritonavir in the neonate and developing infant. which would be a boon because it has all these toxic liabilities associated with it. And this is recapitulated in a more relevant model here in neonatal and adult HLMs. In adult HLMs, in the absence of ritonavir, of course, lopinavir gets cleared very quickly, but not so quickly in pediatric neonatal HLMs. Okay, so this really was the first inkling that clued us in that these antivirals could be an important source of these drug-hormone interactions. I'd like to point out Hannah Work, who's also our ASPET Junior Communication Liaison. She's a PhD candidate in my lab. Has done a lot of excellent work. Graduating next spring. For those of you out there that may be hiring, she would be an excellent candidate. So you can contact her or me about that. But what she did was she screened a library of antiviral compounds, antiviral drugs, I should say, from different classes, including polymerase inhibitors, protease inhibitors, integrase inhibitors, and NRTIs. And what was interesting about this study was that most of the compounds tested were substantial inhibitors of DHEAS hydroxylation. And by substantial, I mean they inhibited greater than 50% of activity at a concentration of 20 micromolar. And again, that's in the relevant range for a lot of these compounds, and that you would find them, in terms of the circulating plasma levels. She went on to characterize them further, some of the more potent inhibitors, by doing IC50 studies, so she could compare paratiprevir, which I'm going to talk about more in a little bit, efavirenz and imifinavir, were all in the 10 to 20 micromolar range. So this is important because the KM for DHEAS metabolism for CYP3A7 has been reported to be from 5 to 20 micromolar. So it's right in that range that you would expect to see inhibition of these compounds in a clinical setting. One thing that jumped out at us right away was HCV antivirals, because they were really significant inhibitors of CYP3A7 metabolism in our assay. And in fact, 8 of the 13 antivirals tested inhibited CYP3A7 activity by 50% or more. And what's important about this is that 3 of these have been used in pregnant people already, okay? And 7 of them have been reported to be CYP3A4 substrates or inhibitors. So we thought that they would have an effect on CYP3A7 as well. So again, she did some IC50 determinations with DHEAS as a substrate for all these HCV inhibitors. And the IC50s range from 20 to 10 well, at least down to 11 micromolar in the case of paratiprevir. So again, in that range, they could be clinically significant. So since paratiprevir was the most potent inhibitor, we decided to follow up on that. And because we had some previous information, well, sorry, I'm getting a little ahead of myself. Now, let me tell you a little bit about paratiprevir first. So this is a drug that was first marketed in 2014. It's an acyl sulfonamide inhibitor of the NS3-4A viral serine protease, so a protease inhibitor. It's reported to be metabolized by CYP3A4. And importantly, it's always dosed with the pharmacoenhancer ritonavir. So you can be having a synergistic effect here where you see inhibition from ritonavir and inhibition from paratiprevir as well. It was withdrawn from the market in 2018. Now, if you go into the literature and read why it was withdrawn, AbbVie says it was withdrawn not based on any sort of toxicity or efficacy issues associated with the compound, but for other reasons. It does, however, cause hepatotoxicity. And it's a rather large drug. You can see here the molecular weight's around 766 Daltons, so a fairly large drug substrate for CYP3A. So based on some of our early data, Hannah decided she would look to see if it was a mechanism-based inactivator of CYP3A7 metabolism. So she initially did an IC50 shift assay. So what we're showing you here is the IC50 shift for paratiprevir on the left and ritonavir on the right. So we start out with a 0-minute pre-incubation, so a normal IC50, and then we do a pre-incubation with NADPH for 30 minutes to see if you're generating reactive metabolite that could potentially inhibit the enzyme. And that's going to lead to a more potent inhibition than what you would see without that incubation. And in fact, she does see an IC50 shift, both for ritonavir and paratiprevir. Of course, it was more significant for ritonavir. I've already told you that's a really potent inhibitor of CYP3A7. So the shift there is almost 4.7. Compare that to paratiprevir, where it's just above 2. Well, 2 is our cutoff for these IC50 shifts in terms of potential clinical effect. So we still think it was an important potential inhibitor in vivo as well. So she followed up with doing both Ki and Kinact studies. So the Ki, she determined for paratiprevir inhibition of DHES metabolism by CYP3A7 was 4.66 micromolar, and the Kinact was 0.00954. So this tells us that it's a moderately potent inhibitor of CYP3A7 metabolism of DHES. Keep in mind, though, it's always dosed with ritonavir. So there's a synergistic effect, again, between the two drugs in terms of inhibition of CYP3A7. So we were fortunate enough to have this crystal structure that the Scott lab generated for us. So we decided to do some docking studies with paratiprevir. So I'm showing you here a model of CYP3A7. We've removed DHES from the active site, and we've docked paratiprevir in the active site using AutoDock-Bina version 1.2. It's shown here in purple. And you can see that phenanthrene ring sits right above the heme iron. So it's likely that we'll probably see some oxidation there, or perhaps an OD alkylation event, liberating the phenanthrene ring totally, which is very fast, energetically facile reaction for CYP3A enzymes to perform. Now, we don't have any metabolism data at this point, and unfortunately, the literature has a dearth of metabolism data in on this drug for CYP3A4 as well. So we don't really know what the metabolites are yet. That's going to be the next step, but at least this gives us some idea of what's happening structurally. So the further questions we have regarding HCV antiviral drug inhibition by CYP3A7 are, do the reactive or potential reactive metabolites that we think are occurring because we see mechanism-based inactivation, do they play a role in this paratiprevir hepatotoxicity? And also, is the observed hepatotoxicity idiosyncratic to each drug? I should mention at this point, more than one drug, HCV antiviral, has been withdrawn from the market and has shown hepatotoxicity. So are they idiosyncratic to each individual drug, or is it conserved across the class? And this is important because if it's conserved across the class, it may point to certain structural alerts that we can modify in this drug class to make them less toxic and more effective. Finally, is that CYP3A7 inhibition observed solely from paratiprevir, the stuff that we see solely from paratiprevir, is that relevant in vivo? Because remember, it's dosed with ritonavir. So ritonavir could potentially be swamping out any effect that we're seeing directly from paratiprevir. That we need to tease out. Okay, I don't know how I'm doing on time here, Hannah. Getting there, okay. The last thing I wanted to talk about was other potential pathways for disruption of that fetal placental communication conduit, specifically with bile acid metabolism. So bile acids are critical for absorption of dietary fats and fat-soluble vitamins. We're all pretty familiar with that. Of course, they're also important signaling molecules. In the last 10 to 15 years, there's a lot of data that's come out to support the importance of these bile acids, which are steroids, as signaling molecules. And CYP3A enzymes are a major metabolic pathway for bile acids in the liver, and thus have an important function in bile acid homeostasis. And this is particularly true in the neonate and developing infant, because bile acid by CYP3A7 is really important in those first few weeks of life due to the fact that the neonatal gut is yet to be colonized by enteric bacteria. So adults have enteric bacteria. That's a major pathway for bile acid metabolism and elimination. A newborn baby doesn't have much enteric bacteria. So it's really relying on CYP3A7 to get rid of toxic levels of lithocoic acid and some of these other bile acids. And some clinical studies have observed limited weight gain in HIV-exposed uninfected infants that were receiving Calatra. And this could also pose an increased risk for neonatal cholestasis, which has also been seen in this cohort. So one of the projects going on in the lab, Sylvie Candle was working on, was looking at bile acid metabolism inhibition by some of these drug cocktails. And she started looking at Calatra, since we already had some data on that. So that's the lopinavir-ritonavir cocktail. And you see at the top panel here, she's looking at oxidation by CYP3A4 of lithocoic acid and quinodeoxycholic acid as well. And you can see it's a little hard to see, but essentially we see complete inhibition at physiologically relevant concentrations of that lopinavir-ritonavir cocktail of both LCA metabolism and CDCA metabolism by CYP3A4. CYP3A7, the effect's not as profound, but it's still very significant. You can see the red line here represents metabolism in the presence of the inhibitors versus the absence, the black line there. probably more significantly, what we did was these experiments with pediatric human liver microsomes, and even in pediatric human liver microsomes, there's dramatic effect, both with 1 week old human liver microsomes, where we saw a dramatic decrease in bile acid metabolism in the presence of chelatora versus the absence, and also in 8-month-old pediatric human liver microsomes. And that's important because this effect can be prolonged. It seems like as long as the patients are on these drugs, you can get suppression of bile acid metabolism. And so that can lead to effects later that are exhibited later in life in terms of dyslipidemias, obesity, and other issues associated with bile acid inhibition. Okay, so just to summarize then, CYP3A7 is an important fetal placental communication conduit during pregnancy. Antivirals from diverse classes can inhibit CYP3A7-mediated metabolism of DHEAS, very important hormone. And HCV antivirals tend to be more potent than some of the others, and they're also dosed during pregnancy, at least some of them have been. Paratiprevir, an HCV protease inhibitor, is a mechanism-based inactivator of CYP3A7. This possibly might be related to its hepatotoxicity. The jury's out on that. That's something we're still looking at. Inhibition of CYP3A7 can also affect bile acid homeostasis in developing neonate, possibly leading to obesity later in life, another avenue we're exploring right now. So hopefully I've been able to show you this little talk. Potential drug-hormone interactions need to be taken into account when prescribing drugs in neonates and pregnant persons in order to protect the fetus and developing infant from unwanted drug toxicity. All right, so I'd just like to acknowledge all the real people in the lab who, well, I'm a real person, but I don't do lab work, right? So the people that are actually doing the work, including Hannah Work, my PhD student in the lab who's going to be graduating soon, Dr. Silly Candle, my better half, partner in science and life, and here she is on a recent ski trip. We like to ski in Colorado, of course. My collaborator, Dr. Emily Scott, over the past few years, who really worked diligently at getting that crystal structure 3A7, and it was pretty exciting to see that come out this year. And then, of course, the funding sources, both R1 from NIAID, R21 from NIEHS, the CU Startup Package, and then previous funding as well. Here's my lab at CU Anschutz. And the last thing I want to talk about before I go, I know this is a ways out, but we have a conference coming up on cytochrome P450 enzymes in 2027. So you can mark your calendars now. It's only 4 years away, but we're very excited about it. It's a conference that rotates between here and Europe and Asia. And the next one's going to be in Switzerland in 2025. Emmett Pandy's chairing that. But I've been very fortunate to assemble awesome A-team group of local organizing committee, including Clarissa Jackson, Simone Brixo-Sandurco, and Fernando Estrada. So all of those people are probably pretty familiar to you. So I hope to see you in Colorado. We've got mountains, beer, and of course, cytochrome P450 if you want to learn more about them. So with that, I'll end and take any questions.
Thank you very much, Jed. That was really a wonderful overview of your current research activities. I know we don't have too much time, so I want to get right to the questions. So there are a few that have been submitted, and I'll just read them off quick, starting with Aiming Yu. Aiming is wondering to what degree the mechanism-based inhibition of paratiprevir would affect the exposure to CYP3A7 substrate drugs / steroids according to the KN activation and KI values you determined.
I love that question. I mean, awesome. I don't have an answer for you, unfortunately. That's something we need to look at. Absolutely. I think it could be important for a DHEAS. I do. Because I think with DHEAS, you're reaching that tipping point, particularly when it's dosed with ritonavir, right? We already know that that's a problem. And so anything that's going to push it a little bit farther so you get less DHEAS oxidation is going to lower those estriol levels in placenta and potentially lead to preterm birth. In terms of other drugs, that's a great question and something I will look at. I should talk to you more about that after. Great question.
Great. Okay, thanks, Jed. The second question is from Chuck Locuson. And his question is the Calatra drug product an equimolar or an equimilligram mixture? I was wondering how you test the mixture in vitro studies.
So Sylvie Candle is the one in my lab doing that. I believe, if I remember correctly, it was equimolar, and maybe Hannah can correct me if I'm wrong. It's equimolar, and we tested at the concentrations that you would expect to find, the circulating concentration. So for ritonavir, it's nanomolar. It's pretty low, but it's a pretty potent inhibitor.
Oh, okay, there's one more question, and this comes from Josh Yu. And Josh asked, does lopinavir and ritonavir get through the placenta?
Thanks, Josh. That is another great question. And you're keeping me honest. You were paying attention. Yeah, we don't know. We don't know how much gets across the placenta. And it's really hard to have any answer for that, at least the human placenta, right? I think that's something something else we need to look into. Modeling is difficult, but that's about the only way you can kind of get at what the answer is, how much of this is getting across the placenta and affecting the fetus. Thanks for that. That was a good question.
Great, Jed. Just one final comment from me. I think the paritaprevir hepatotoxicity thing is interesting. And I'm wondering, like with HCV, with infection, we often have a depression of cytochrome P450 enzymes.
Yes.
And so I think you wonder if, you're treating hepatitis C and you're getting a resolution of the infection, and maybe now you start seeing an increase in CYP activity, and maybe you actually get more, if there's reactive metabolites being produced, more occurring as the infection resolves, and perhaps that could cause an issue. Anyway, that not just kind of hypothesizing, but would be interesting to think about that.
That's very interesting, Carrie. So a rebound effect where you're actually producing more of the reactive metabolites, you're getting more metabolism. That's really interesting. Yeah, for sure.
We have one more question actually came through the chat.
Oh,
not
okay.
the Q&A.
Oh, okay. Sorry, I didn't see that.
Did you see that one?
Try to answer really quick. I don't want to take too much of Colton's time. Yeah, we need to move on to Colton's talk. So can I answer real quick, like one sentence?
Yeah, can you see the question, Jed?
Okay, question. Yeah. There are some plant products or their metabolites that may affect metabolism through activating this enzyme. Can you comment on it? I'm not extremely versed in that area. I mean, certainly that's true of the other 3A enzymes that activate metabolism, flavones, alpha-naphtoflavone. So it could be the case here as well. And I'm going to thank you for that question, but I'm going to end there because I'm taking too much of Colton's time. So thanks.
Okay, great. So I will turn it on to Julie, who's going to introduce our next speaker. Thank you. Thanks, Jed.
Thank you, Jed. Thanks, Carrie. So our next speaker, Yolan, I believe you have a slide perhaps you can pull up. If not, we'll go without the slide. But our next speaker is Colton Eberhard. He's a graduate student at Johns Hopkins University School of Medicine in the Department of Pharmacology and Molecular Sciences. And he's been researching under the advisor, Dr. Namanje Bumpus, investigating bioactivation of tenofovir and looking at the role of creatine kinases. Also, one other thing I wanted to mention is Colton has won the Graduate Student Poster Award 2 years in a row at ASPET. So great work, Colton. I'm looking forward to your talk.
All right, thank you, Julie. And thank you to the organizers for having me. It's an honor to be asked to give a presentation for this seminar. Like Julie said, I'll be giving a brief overview of my thesis research that has been focused on the role of creatine kinase brain type in tenofovir metabolism and activation. So a brief overview, I'll give you a little introduction into the tenofovir activation pathway, and then go into how we have been assessing the activation of tenofovir in vitro, and then also showing you a novel CKB knockout mouse model that we have generated and are using to understand CKB-mediated tenofovir activation in vivo. And then lastly, I'll end off with an overview of some of the proteomic studies that we're doing to assess the changes that are occurring in these knockout mice. So as I'm sure a lot of you are aware, nucleotide analogs are a major class of antiviral drugs. The antiviral drug that I'm specifically studying is tenofovir, which is used to treat and prevent HIV infection, as well as chronic hepatitis B virus infection. And like most nucleotide analogs, the active metabolite is way too hydrophilic to cross any barrier. So it's actually formulated as two different prodrugs, tenofovir disoproxyl fumarate or tenofovir alafenamide. In each case, the two protection groups on this phosphonate moiety have to be cleaved or removed to provide the tenofovir parent drug. This then has to undergo two sequential phosphorylations to become pharmacologically active. And this metabolite here mimics ATP in structure and is incorporated into growing DNA strands, causing chain elongation pause. So while tenofovir efficacy remains high, the clinical outcomes can be heterogeneous. And this is often seen with tenofovir-based pre-exposure prophylaxis for HIV, or we call it PrEP. And a lot of the earlier clinical studies have attributed this variability in efficacy to a lack of adherence. So about a decade ago, studies started employing dried blood spot analyses where they would take blood spots from individuals in these trials and detect the level of tenofovir diphosphate in the blood, and then correlate that directly to adherence or doses taken per week, which essentially negated any bias by self-reporting on adherence. Unfortunately, even high adherence groups were still showing treatment failure. So for example, a recent study from 2022 investigating PrEP efficacy in adolescent girls and young women showed 16 seroconversions, which is a pretty, it had pretty high efficacy with only 2.2 incidences per 100 person years. But diving deeper into the actual data, we saw that 8 of those individuals had detectable levels of the active metabolite in their blood, and 3 individuals had enough to equate to 4 or more doses taken per week, which would be the cutoff for high adherence. Additionally, in the last 5 years or so, there have been 2 case reports talking about 2 individuals taking tenofovir-based PrEP, maintaining high adherence based off of dried blood spot analyses, and they still ended up acquiring a tenofovir susceptible HIV strain. So why are these treatments failing? Our lab has been investigating this for quite some time now, and a study by Julie Lade in 2015 showed that each compartment has a slightly different activation pathway. So for example, in PBMCs in vaginal tissue, the adenylate kinase is responsible for the first activation step, but then the second activation step is completed by the pyruvate kinase isoenzymes. On the other hand, the colon tissue, while adenylate kinase completes that first reaction, the second reaction is mainly completed by creatine kinase muscle type. So these were 3 different tissues that were susceptible to HIV infection. However, with increasing knowledge on how HIV kind of harbors in certain tissues, we had no idea the enzymes responsible for catalyzing tenofovir activation in the brain or in the liver. And so my PhD project has set out to identify some of these enzymes. And using creatine kinase muscle type as a springboard, we chose to look at the other creatine kinase family members and see how they may be contributing to tenofovir activation and in what tissues they might be helping. So after we came back from COVID lockdowns, my first experiment in the lab was to run this assay in vitro, where I incubated recombinantly expressed and purified creatine kinase isoenzymes in the presence of the tenofovir monophosphate metabolite and the necessary substrate and then detected the formation of tenofovir diphosphate using ultra high performance liquid chromatography, tandem mass spectrometry. And what we found was there was no significant change between the isoenzymes, which was kind of interesting to us, given that this mitochondrial isoform actually does the opposite reverse reaction. And so looking at whether there was intrinsic differences between the three enzymes, we looked at the canonical reaction, which is the formation of ATP, and saw again that there were no changes in ATP formation between any of those enzymes. And so with this, we had to narrow it down a little bit more, and I decided to choose CKB to investigate further, because some early on preliminary proteomics data showed that CKB was in the top 25 most abundant proteins in the brain, and RNA data showed that it was highly expressed in astrocytes, which is a specific brain type, right? The specific cell type in the brain that harbors HIV infection. And so using CKB, using this information about CKB, we decided to move forward with this as a candidate to identify if it is activating tenofovir. And going back to why are we even interested in this, we believe that inter-individual variability in the activation of tenofovir could be leading to this efficacy variability. And that can be explained by identifying the enzymes responsible for activation, but also seeing how mutations disrupt that activity. And so what I had done is recombinantly expressed and purified 15 naturally occurring mutations in CKB, and found this pretty stark trend where mutations in the active site essentially had no activity toward forming tenofovir diphosphate, and then slowly going back to the external surface with activity increasing. Of note, only one mutation in the dimer interface caused a significant reduction in the formation of tenofovir diphosphate, and we showed that it's not necessarily residue specific, but mutation specific, where this arginine 172 to a glycine had little to no impact, but the same residue mutated to a proline had significant reduction. We see this down here with the histidine 296 mutations as well. Again, looking at whether these have intrinsic changes with the canonical reaction, these 5 mutations here showed no significant change in the canonical reaction, which would be the ATP formation, indicating that individuals who possess mutations at these sites may not necessarily have a typical pathophysiological phenotype associated with decreases in CKB function, but they might not be able to activate tenofovir to the extent as other individuals. So with all of that in vitro data, we still didn't really know where CKB could be contributing to tenofovir activation. And so we decided to make a CKB knockout mouse model. And with the help of the Johns Hopkins transgenic core, we targeted the CKB gene in C57 black 6 mice with 2 guide RNAs. Interestingly, the most productive male breeder actually possessed 2 mutations on the same allele, one 55 base pair deletion that encompassed the start codon, and then a 28 base pair deletion that was about halfway down exon 2, which is the first coding exon. We extracted mRNA from knockout or homozygote brains and sequenced the cDNA, and found that there's actually a new splice acceptor, which I've highlighted here in red, that generates a frame shift in a new start codon, leading to, if translated, a mRNA sequence that would provide only 5 amino acids and then a stop codon. So we were pretty confident that this would lead to protein knockout, and use this mutation to generate our CKB knockout mouse strain. And then what I'm showing here is just a simple immunoblot showing that homozygote males and females had no CKB present in their brain. So next, we needed to determine whether tenofovir activation was still occurring in brains that lacked CKB. And to do that, we used brain lysates that had active protein still in the very similar in vitro assay where we incubate the tenofovir monophosphate metabolite with the brain lysate and then 3 common phosphodonors. So we decided on choosing these phosphodonors, ATP because it's the universal phosphate donor, phosphoenolpyruvate because we knew that pyruvate kinase enzymes need that as a substrate in order to activate tenofovir, and then lastly phosphocreatine, which would be the creatine kinase substrate. And then again, we detected the formation of tenofovir diphosphate by HPLC mass spectrometry. And what we see is pretty dramatic. The knockout animals had significantly less tenofovir diphosphate formed in brain lysates than the wild type animals. So for example, the knockout males had 70% less tenofovir diphosphate formed. The knockout females had 77% less formed. But also of interest, the sexual dimorphism here between the wild type male and the wild type female, where the wild type female had 45% less tenofovir diphosphate formation. So from this data, while it is in vitro and we can't conclude about membranes or transporters or anything like that, this data provides evidence that CKB is likely one of the main enzymes contributing to tenofovir activation in the brain, and that there is some sort of sexual dimorphism happening in this bioactivation pathway. So next, we wanted to know how in vivo dosing would affect the activation of tenofovir. And so we dosed homozygote and wild type mice for 2 weeks with a combination therapy. This is used most common for the prevention of HIV infection. It's tenofovir disoproxyl fumarate in combination with M-tricytidine. And then the tenofovir metabolites were extracted from each tissue and run on LCMS. What I'm showing here is our method for tenofovir detection. We were able to simultaneously detect tenofovir and tenofovir diphosphate. And in order to determine tenofovir activation, we use the peak area under the curve as a function of that metabolite concentration. And specifically, the activation would then be the diphosphate area divided by the tenofovir parent area. And what we find is that in knockout males, there's a slight reduction in the tenofovir activation in liver, only about 22%. But then again, we see this sexual dimorphism between wild type males and wild type females, and that's about 36%. This data here suggests that CKB could be contributing to activation in the liver, but likely that other enzymes are compensating for the loss, resulting in this not being a complete dramatic reduction like we saw in the brain. So next, we decided to look at the proteomics, the proteome of the brain and the liver tissue to get a better idea of what was happening at the proteomic level. And to do that, we use data-independent acquisition, which essentially we can extract proteins from tissue, we digest them with trypsin, quantify them, and inject the same amount of peptide per run. We use a nano LC system with a timsTOF mass spectrometer. And the DIA method is the most robust method in identifying proteins. And this is because essentially everything in your sample is fragmented. And that's done by using isolation windows. So the first scan would be an MS1 and would detect a wide range of what we call precursor ions. And then the subsequent runs would isolate smaller M over Z ranges, and everything would be fragmented, giving many more spectra to identify everything in the sample. So we use computational methods to essentially sequence every peptide that is injected. And so to go back to what we're seeing in the brain, the formation of tenofovir diphosphate, we saw a reduction in the knockouts, but also in the female wild types. And we would think that this would only be affecting the final phosphorylation event. This is shown here. Again, the CKB knockouts have no CKB in them, as confirmed by our immunoblot earlier. But we also see, interestingly, that other enzymes known to activate tenofovir are also differentially abundant. So for example, CKM, which I mentioned earlier as contributing to activation in the colon, we see significant reduction in the knockouts. And then in the pyruvate kinase muscle isoenzyme, we see a reduction in the knockouts in this strange sexual dimorphism between the male and female wild types. We also looked at enzymes at other parts of the tenofovir regulation pathway. And interestingly, this last regulation where tenofovir diphosphate is converted back to tenofovir biphosphatase, we see the one enzyme that has been published on for having that activity, we see an increase in abundance in that knockout, indicating that differentially abundant proteins in this pathway could all be compounding, causing this formation of tenofovir diphosphate to be reduced. So then looking at the liver, which is a little bit more peculiar, we were unable to detect any creatine kinase enzyme in the liver, which was shocking at first because we saw this reduction in our knockout animals. But thinking back to what we saw in the brain, we started looking at the entire activation pathway and how the knockout could be affecting the abundance of those enzymes. And so the first reaction here, as I mentioned earlier, is completed by adenylate kinases. And we see that in knockouts, they're significantly increased, but we do see the sexual dimorphism here with the wild type male and the wild type female, where they're significantly reduced. And then this last reaction done by the pyruvate kinase enzymes, again, they're differentially abundant between genotypes and sexes, with, in addition to the other tenofovir activating enzymes, maybe compounding, resulting in this effect on the pharmacology of tenofovir. So in conclusion, I've shown that CKB can phosphorylate tenofovir monophosphate to the active metabolite, and that mutations in CKB can decrease that activity depending on where it's located and the specific residue it's being mutated to. I've shown that the in-brain lysates of knockout mice, tenofovir diphosphate formation is diminished, and that tenofovir activation in knockout male livers is reduced. Our proteomics data showed that nucleotide and small molecule kinases with known tenofovir activity are differentially abundant, which could lead to a compounded effect, forming the active metabolites, and that there is sexual dimorphism present in this activation pathway. And that was observed in both liver and brain of the wild type mice. All right, and then with that, I would like to thank my lab and my advisors, Namanje and Ben, and especially Eric Mosher for helping a lot with the in vitro assays early on in the project. I'd also like to thank my thesis committee for keeping me on track and getting me to the goal of graduating. And then lastly, our funding. And I can take any questions.
Great presentation, Colton. Thank you so much. We have a few questions in the Q&A and in the chat. The first question is from Savita Sharma. The question is, what kind of column did you use for the LCMS method development that you showed? And then what was the second peak in the tenofovir at 7.05 minutes?
Yeah, so the column that we're using is a Halo C18 column. We actually are using an ion pairing agent to help with the separation of the metabolites. And that second peak in the tenofovir chromatogram is the tenofovir monophosphate metabolite that is decaying in source. One of those phosphates seems to pop off quite a bit. And so we see that with the detection of the tenofovir M over Z.
Great, thank you. The next question is, what effect do you think genetic variation in the CES enzyme complement has? That question's from Jed Lamp.
Yeah, so we think that it could very well be a factor, especially with TDF, where a lot of those protection groups are cleaved before the drug even enters the cell. So that's another factor that we should be addressing as well. There are groups studying that. We are more interested in the last step because essentially that's the one that would get you the metabolite. But yeah, we do see that anywhere in that entire pathway, any inter-individual variability could be causing these treatment failures.
Great, thank you. Another question is curious why the FTC was co-administered in the mice, and do we anticipate concurrent FTC metabolism to have potentially affected tenofovir metabolism?
Yeah, so we used emtricitabine because it's most oftenly, tenofovir is most oftenly prescribed as a combination therapy with emtricitabine. So we would have a better idea of if there are some type of drug-drug interactions there. One thing that we noticed when doing our proteomic analysis was that enzymes known to activate emtricitabine as well are differentially abundant. Interestingly, though, they are higher in abundance in the knockouts than in the wild type. So we don't think that that would impact emtricitabine negatively. If anything, it would help kind of push emtricitabine along that pathway. But yeah, I think any interaction between the two drugs, if loss of CKB affects emtricitabine as well, are things that need to be followed up on.
Thank you. That question was from Elaine. One more question here. Do we know if your observed sexual dimorphisms in the knockout mouse model are conserved in humans as well?
Yeah, that's one of our big questions. We're not really sure. There's been a lot of clinical trials looking at tenofovir efficacy in women, and they seem to be not as great as in males. There are several confounding factors there, but we wonder too if that could be because of CKB or some of these other enzymes in that pathway just being differentially abundant in these tissues susceptible to HIV infection.
Great, thank you. That question was from Julie Lade. Thanks so much, Colton. I'm going to hand it back over to Young. Thank you.
Okay, thank you so much. Those were two great talks. So we got to learn about the hormone-drug interactions in pregnancy and also the enzymes involved in tenofovir bioactivation. Those were all very great, good talks. With this, we conclude our presentation, the talks, and I will hand it over to Yolan.
Thank you, Young, and thank you all for joining us today. Please be sure to complete our survey when you exit the Zoom session. A recording of today's session will be posted in Aspid Connect next week. Please be sure to check that in there. The Aspid 2024 annual meeting will be held in Arlington, Virginia, May 16th to the 19th. The September 29th to submit a proposal is rapidly approaching. So you can scan the code on the slide so you can learn how to submit. We have exciting focus sessions coming up in the fall. Visit the link in the chat to bookmark our schedule. And thanks again to the speakers and organizers. Another great session. Thank you guys and have a great week, everybody. Bye-bye.